Welcome to this platform. So we continue with revisions. I hope you've looked at or you've seen paper 1, that is 2023. So this is paper 2, 2023, biology, that is for GCE. Okay, so let us look at the questions that came. We are going to revise only section A. So here is the first question that we are going to look at. And the question reads, Figure 1 shows different types of specialized cells. As you can see, the cells A, B, and C. The first question, identify cells A, B, and C. Identify the cells A, B, and C. So for cell A is nerve cell. Then of course cell B, root hair cell. Then we have C, ciliated cells. Okay, so these are cells that we have, that is A, B, C. Then of course the question 2 of A, state the parts of living organisms in which specialized cells B and C are found. So where is B and C found? So for B is found in the roots. Then of course ciliated cells, 1, they can be found in the oviduct or respiratory tract. So this is where you find the ciliated cells. So any of these, you should be given a mark. Oviduct or fallopian tube, respiratory tract, and in each I'm sure we're going to look at the functions. What do they do there? Explain the functions of cells in figure 1. Explain the functions of each cell in figure 1. So we are giving the functions of these. So the first one, nerve cell, what is the function? So conduct electric impulse. So you can say conduct electrical impulse. Conduct electrical impulse or nerve impulse from one part of the body to another. Now here we haven't specified whether we are dealing with because these are in 3 types. We have the sensory, we have the motor, we have the relay. So here we're just giving generally. Okay, then of course B, since we are looking at the root hair cell, so one of the function is absorption for the root hair cell, absorption of water. So absorption of water and mineral salts. Also we can put anchors the soil, anchors the plant into the soil. Anchors the plant into the soil. We should be given a mark N of these. Then what about C, which is the ciliated cell? So C, we said, is found in the respiratory tract and of course oviduct. So the first function I'll give is for the function in the respiratory tract. So I'll say in the respiratory tract, they sweep out mucus, containing dust and germs. Okay, what about in the oviduct? So in the oviduct, I would say in the oviduct, they move the ovum. They move the ovum towards the uterus. Okay, so these are the functions. So that is it all about question 1. Then what about question 2? Figure 2 represents the rhizopus growing on bread. So this question also came in 2023 4, but of course different questions. The first question is identify the part labeled E, F, and G. So what are the parts labeled E, F, and G? So for E is called the sporangiopho. Then of course we have F, which is the rhizoid. Then of course we have G, which are spores. So these are the parts. State the type of nutrition found in this or in organism in figure 2. So the type of nutrition is called saprophytic nutrition. So saprophytic nutrition, where the organisms feed on already dead decaying matters. Explain how the nutrition identified in above is brought about. So how is saprophytic nutrition carried out or brought about? That is very important to understand. So we can say organisms that feed saprophytically, by the way, they are called saprophytes. So we can say saprophytes feed by secreting extracellular enzymes, or you can put extracellular digestive enzymes, okay, from the hypha, which are rhizoid. So hypha or rhizoid, then full stop. The enzymes hydrolyzes, okay, hydrolyzes substrate. You know that the substance that is hydrolyzed called substrate externally. So they hydrolyze the substrate externally, then the end products are absorbed. The end product are absorbed internally. So I'm just going even up to this space. Are absorbed internally by the rhizoid or the feeding hypha and assimilated, okay, assimilated internally. Okay, so the saprophytes feeds by secreting extracellular digestive enzymes from the hypha or rhizoid. The enzyme hydrolyzes the substrate externally, then the end product are absorbed internally by the rhizoid and assimilated internally. Okay, so that is how it is brought about. And of course, if the substrate are carbohydrates, enzymes hydrolyze the carbohydrate into glucose. Then of course, if you are looking at proteins, proteins are broken down to amino acids. If you are talking of lipids, they are broken to acid, fat acid, and glycerol, and now absorbed and assimilated internally. Internally, it means inside the organisms. Okay, so I guess you've gotten that. Now, this space, we'll leave it for the other question. So let me just, I don't know if I can remove this one. Let me remove this one and of course read this question. Describe the importance of the importance of nutrition in the rhizopus to the environment. What is the importance to the environment? So focus on the environment. It's very important to understand. I know you have a lot of importance of saprophyte, but this is specifically to the environment. So 1, used in what? Recycling of nutrients. Okay, used in recycling of nutrients, mainly because they act on decay matter and the nutrients are recycled back into the soil. Then also used in eradicating. Okay, so used in eradication of dead matters. So it is used in eradicating of dead matters or dead bodies, to get rid of dead bodies. Just imagine if there are no saprophytes, you find that let's say dogs die, they will continue just accumulating or piling without decaying. Okay, Aside from that, it is used also in treatment of sewage. Then also you can talk of use as food, but of course not related to the environment. Okay, so they are also used as the food. In the case of mushroom, mushroom is a saprophyte. I guess you knew that. So that is the end of question 2. We go to question 3. So for question 3, the question came from that is respiration in fish, or respiratory organs, that is in fish. So figure 3 shows part of respiratory organs of a bone fish. Identify the structure Z. So what are the structures Z? So you remember this question, we found it also in multiple choice, but of course it's in different form. So here the structures Z are called gill filaments. Okay, gill filaments. Now, explain how the structures Z are suitable for the function. So in short, they are asking us to give the adaptation of gill filaments. So they are numerous in number. Okay, so you can say they are numerous. They are numerous to provide the large surface area. Provide the large surface area for diffusion. For diffusion of gases. Okay, look at this. These that you are seeing, they're all filaments. You've seen they're a lot in number. In short, they are numerous in number, and that increases the surface area for the gaseous exchange. Then now question 3 of A. Name 2 other respiratory organs in a fish other than the one shown. So one, you can talk of the other one, we can talk of the operculum. You can talk of we can talk of also the mouth. Yes, the mouth. Now the other question is describe the role played by each of the respiratory organ identified. So the one that we've mentioned first is the operculum. What is the function of the operculum? So the operculum covers on the gills. So simply protects the what? Protects the gills from external harm. So protects the gill from external harm. Okay, then of course, what about the mouth? What is the function of the mouth? So the mouth is the one that opens. You know that the fish breathes through the mouth, though it breathes by taking in water. So the mouth, we can say it takes in water rich in oxygen and pump it over gills. Okay, so that is what we can say in terms of the function. So please be able to bring out the other functions that you think or are the functions to this. I would like to hear from you as well. Which respiratory organs in human plays the same role as the one or as the structure in Z in a bone fish? Which respiratory organ in humans plays the same role as structure Z in the bone fish? So now we are just looking at if it was the whole of a gill, we are going to say lungs. But here, for me, I feel they are just talking of Z, and Z here is for gaseous exchange. So in other words, they are asking where in humans does gaseous exchange occur? So it occurs in the what? In the ovulus or ovuli. A lot of them are called ovuli. So please be able to also look at this. That is my understanding of this question. Otherwise, we are not going to write lungs because we're just looking at structure Z. Okay, up to question 4. So the figure 4 shows the relationship That is shows the relationship between blood, tissue, and that is blood, tissue fluid, cells, and lymph. Okay. What is the first question? Identify identify U and T. So U is right here, which is the lymphatic. This is called lymphatic vessel. So U is the lymphatic vessel. Okay. Then of course, what about T? T is the venue. So the end part of the vein is called the venue. Okay. And of course, when it comes here, this now become the capillary. The capillary is right here forming the capillary bed. Name one nutrient represented by S. So we are simply looking at the nutrient that is filtered out. So when the blood is coming here, you know that blood, as it being transported from the heart, it contains nutrients, oxygen, and all that. So when it reaches here, the capillaries have porous membrane, I should say. So due to high pressure here, it filters out. So the tissue fluid, the plasma filters out, or it is pushed into this surface here, or this part. And of course, it becomes the tissue fluid. And of course, in the tissue fluid, that is where you find, here, this is where you find the cells. And these cells need oxygen. So one of the nutrients that you find there is needs oxygen and glucose. So the nutrient that we find there is what? Glucose. Compare the blood pressure at R and that of T. Give a reason for your answer. So what is the difference between pressure at R and T? So R is called arterial end or artery. Then of course this one is called venue or vein. So artery and vein, in terms of their structure, is that, so the reason is the pressure. So you can say the pressure of blood coming from the arterioles, is higher than the pressure in the venue. Okay, so, or in other words, you can say the pressure in the arterial is higher than the pressure in the venue, but we are looking at it in terms of blood. So the pressure of blood coming from the arterioles, or should have just said the pressure in the arterial is higher than the pressure in the venue. I guess that would be okay. Now, what is the reason? The reason has to do with their structure. So because the arterioles or arteries have relatively, I'll put the word relatively thick, so relatively thick muscular walls than the what? The venue or the veins. So that is the reason why it has a relative thick walls than the venues. And the reason is because it carries blood at high pressure, and this is is near to the heart. So it carries blood from the heart to all parts of the body, and therefore it moves at high pressure. Now, to withstand that pressure, the design of the arterioles, they have relatively high thick walls. Okay, so what about the other question is that explain the structure U in relation to the blood circulatory system. Explain the role of U in relation to the blood circulatory system. In other words, explain the lymphatic system or lymphatic vessel or lymphatic system in relation to the what? The blood circulatory system. So 3 marks here. So 1, it drains excess tissue fluids and takes it back into the blood. So it drains the excess tissue fluids and take it back into the blood. In other words, it maintains the fluid in the blood. It adds also lymphocyte, so it cleans the tissue fluids. In the case there are some bacteria, it kills all those. So in other words, we are saying it adds lymphocytes. You know that lymphocytes are white blood cells. Lymphocytes to the what? To the blood. You know that lymphocytes are also made along this same lymphatic system. Then also we can say it absorbs and transport. So it absorbs and transport. So absorbs and transport cholesterol, fat, acids, and glycerol to blood. Okay, so these are some of the roles of you in relation to blood circulatory system. Okay, what about question 5? So for question 5 is normally on genetics. So like I said, on genetics, I'll just compile the questions from as far as 2018 to 2024 GCE. So the question is, a hemophilic mother has two sons. A hemophilic mother has two sons who were both hemophilic. The father was non-hemophilic. A hemophilic mother had two sons who were both hemophilic. The father was non-hemophilic. Now, this hemophilia is a sex-linked disease, and of course is recessive. So it is only expressed when you have all the you have two recessive. And of course, you know that in a sex chromosome for male have X and Y, so there's no carrier. It's either you are normal or you are sick. But for female can be a carrier or sick. So this is how it is presented. So define hemophilia. So for hemophilia, we can say this is an inherited disease where a human being breeds for a longer period. Than normal due to lack of clotting factors in what? In blood. So using letter X small and X big to represent the genotype of the father, to represent the genotype of the mother, the father and the mother. So the father, we are saying the father is non-hemophilic. So like I said, you know that the here you have X and Y in male. So if it's non-hemophilic, which means we have the capital letter H, which is just normal, then of course Y there's none. It normally attached to the X chromosome. Then of course for female, for the mother is 6, so we have small h. So small h, small h. So these are the genotype. Then of course, using the genetic diagram, explain why both boys were hemophilic. Okay, so let us close. So we have parents, that is father, and of course mother. Then of course we have parental phenotype. So parental phenotype, we have non-hemophilic. So please finish this. And also we have hemophilic. Then of course we have parental phenotype. So for parental phenotype, we have genotype, parental genotype, genotype. So we have X, H, and of course Y. Then of course this one is X, H, X, H. Then we have parental gametes. Okay, parental gametes. So for parental gametes, we are going to put X, H, Y. Then of course here we have X, H, X, H. Then of course we do there's fertilization taking place here. So you're going to have X, H, X, H. Then of course this and that will have X, H. So start with the dominant. Okay, then we have X, H. Then this one with this one, it will be X, H, Y. Then this and that will be X, H, Y. And of course first generation. So we are saying first offspring, F1 offspring, F1 offspring genotype. Then of course F1 offspring phenotype. So for the phenotype it will be non-hemophilic. This is non-hemophilic but carrier, and also this one non-hemophilic but carrier, but this one is hemophilic, and also this one is what? Hemophilic. Okay, here we can put fertilization. Okay, so that is how we can put up the genetic diagram, and of course this explains why 2 of the sons were hemophilic. Okay, so please, this brings us to the end of the presentation. As always, if you're interested in the online lessons, you can still contact 0976, that is 0976 402563. Let us prepare you for the distinctions. Otherwise, see you in the next one.